and that's going to be from Andy. Andy is from ThinkCyte. Is Andy there?
Yeah. Hi.
Brilliant, Andy. I don't know if your camera's there. There we go. And so ThinkCyte is really interesting because of their AI-based label-free technology for sorting cells. But again, Andy, I'm not going to take anything away from you. I'm going to give you the floor straight away so you can tell us all the details about ThinkCyte. Thank you so much, Andy.
Yep, great. Thank you for the introduction. So let me get started. So ThinkCyte, we are a startup founded in 2016 based in Tokyo. We are currently developing a novel single-cell analysis and sorting platform called Go Cytometry, where the technology was first described and published in Science in 2018. We're currently working with a variety of external partners, including global pharmas and leading research institutes to develop applications in cell therapy, clinical diagnostics, and drug discovery. So as a very brief background, in conventional single-cell analysis and sorting, there's always this trade-off between the amount of content versus the speed. So for example, high-end microscopes give you a lot of information about a cell, but manual manipulation is slow and you can't sort with a microscope. On the other end of the spectrum, conventional label-based cell sorting, such as FACS or MAX, is fast, but you're limited by what the labels or markers can actually tag, and which is mostly single data points. So with Go Cytometry, you can now achieve real-time high-content analysis and sorting of single cells in flow. So our core technology can be summarized as an AI-powered, high-speed image information-based cell analyzer or sorter with both label-free and fluorescence modes. So I'll go through the details here. So, what we have created here is a proprietary optics design that utilizes structured illumination to capture the image information of cells at a subcellular resolution. So this is done without the use of CCD cameras. And this information is recorded and compressed into a 1D waveform, which we call GMI waveforms. So the GMI waveforms are basically a spectral fingerprint of the cell in which we can map or link to specific target phenotypes. And by training using AI, we can develop classifiers to analyze and sort the cells based on the GMI waveforms only. And then we have our sorting mechanism to sort the cells. So our system is capable of multiple modalities, such as looking at diffraction, scattering, and absorption of light. So this really gives us a 3D comprehensive view of the cell. And also we're able to do this in fluorescence mode as well. So there are really two approaches to using Go Cytometry. First is the supervised learning approach, where we run the training samples with ground truth labels through the system, and we acquire the GMI fingerprints for the respective gates. So then with this, we can build a machine learning classifier and use GMI signals only for label-free analysis and sorting in downstream processes. The other approach is unsupervised learning, where we simply run the samples through the system and use AI to analyze their GMI signals to identify unique clusters of interest. So this approach is particularly useful for identifying and discovering subpopulations or subsets of cells where no markers currently exist. Again, our key applications are in cell therapy, clinical diagnostics, and drug discovery. So we have actually demonstrated various proof of concepts with our system where we can distinguish different cell types, including immune cells, cancer cells, and stem cells. But what's more interesting is that our system is also capable of identifying different subtle cellular states or statuses. So things like cell viability, activation, and so on. Another unique kind of feature of our system is that we can quantify at subcellular resolution, allowing for analyzing phenotypes such as nuclear translocation, organelle localization, protein-protein interaction, and so on. And this is currently not possible with conventional technologies. And since Go Cytometry is a flow-based system, we're able to perform phenotypic screening for drug discovery in a pooled format, which can be much faster than traditional arrayed microplates. So this works for both functional genomics and compound screening. So to summarize, the key value propositions of Go Cytometry includes label-free cell sorting, which gives you truly untouched cells and a reduction of cost, intracellular resolution, which gives you the spatial quantification of intracellular phenotypes, and high content and flow. So this allows the large-scale screening and sorting, as well as a novel data modality. So this can be used for novel biomarker and subpopulation discovery for exploratory science. So thank you. And here's a brief company overview with any questions.
Thank you so much, Andy, for another excellent and clear presentation, a pitch around ThinkCyte. There's questions already coming in, so I'm going to jump straight to them. And And I'd like to know, do you have any experience with plant cells or with insect cells? And could your technology be adapted to them?
Yeah, so we've mostly worked with human cells, but we have done with yeast cells, I believe. So, you know, the cells really work the same way between human cells and plant cells or yeast or insect cells and so on. Yeah.
Thank you. And then, is your technology limited to cells? Could you imagine sorting microsomes or organelles? And how small could your selection process be?
Right. So currently the resolution is at a cell level. So the smallest, I think, particle that we work with is around 10 micrometers. So we haven't really tested for anything smaller than that. So we will have to actually try to know. Yeah.
And do you think it could be done with microsomes or organelles, or is that something which is probably too challenging?
It could be a challenge. So right now, again, with the resolution, so there is an option to kind of switch out the resolution with a higher resolution lens. So there could be possibilities there. Yeah, but that needs to be explored.
And what's in terms of geographic focus? Where's your main customer base? What sort of geographies are they in?
Yeah, so currently we're mostly focused in Japan and the US. So we've just started our US expansion mid last year, but by next year we are planning to look into Europe as well as China's.
Okay. And if there's one thing which you'd like to see your technology do in the future, what would it be?
Well, we definitely love to see this in all research labs, because we do believe this is a platform technology that can be used in various places in life science research. And, you know, it is complementary to the current flow cytometry system. So I think there's a lot of use there. Yeah.
Thank you so much, Andy, for your time. Thank you for the fantastic pitch and answering the questions.
Great. Thank you.
Thank you very much.